Hey there everyone. So this is a video on physiology. So in the previous lecture, we had studied the functional organization of the nervous system. As you can already see here, these are the notes that I discussed the complete chapter in detail. So we studied the functional organization of the nervous system, and in today's video, we are going to start with the synapses. Okay, so we are going to see the synaptic transmission, types of synaptic transmissions and other things. So let's get started. So I'm covering this in detail, each and every line from the textbook. So this video is going to form amazing basics for you people, so make sure to watch till the end. And yeah, so okay, synaptic transmission in CNS. There are neuronal junctions where information is passed from one neuron to another neuron. Okay, so there is junction between the the neuron and another neuron. Okay, so from there the transmission occurs. So there are many types of transmissions like chemical transmission, electrical transmission, etc. But in this chapter we are mainly concerned with the chemical transmission. Okay, so they are the neuronal junctions where the information is passed from one neuron to the another neuron. Okay, one neuron to another. So the neural messages are conveyed from PNS to CNS and from CNS to PNS as the target organ. Okay, in this the synaptic transmission is used. Yeah, now so accumulation and alteration in the synapse. Okay, accumulation, wait, okay, accumulation and alteration in the synapse. So what happens is that the synapse, okay, there are a lot of synapses taking place every day. So see, what happens is they learning and processing and memory is also done in the higher centers of the brain. So this is due to the accumulation and alteration in the synapses. Okay, so we are seeing the basic and the general ideas of this first. Now, new synapses and its modifications continue throughout the life of an individual. Okay, so see, there are new synapses occurring, the modifications in the already existing synapses. Because see, what happens is whenever you learn something new, so new synapses is formed, and that helps in learning. Okay, then processing and memory is when you do or repeat the same thing again and again. Suppose you're watching this lecture for the first time, you're learning this for the first time, then your new synapses formed. And suppose you're watching this video for the third time or the fourth time, so that gets into the memory. So this is about the synapse, and it continues throughout the life of an individual. Okay, so these are the things about synapse. And an important point to note that there are innumerate neurons. Okay, there are so many neurons in the brain, and each neuron can generate 2000 synapses. So see, if like there are so many synapses, just multiply and see how much it will be. So 2000 synapses, it is such a huge number. Okay, now according to the number of neurons, so we are going to see the classification first. So according to the number of neurons, see, we have 1 to 1, many to 1, and 1 to many. Wait, I think I missed something. No. Okay, see, we are going to see the types of synapses. Okay, we are going to see the types of synapses. So first we are going to see according to the part of neuron in which the synapse formation takes place. See, we are going to see the neuron in which the synaptic formation is taking place. So see, there are total of 4 types. I have explained with the help of diagram as well. So first one is, it is in the name itself. Okay, it is present in the name itself. So understand the name, the parts of the neurons, the part of the neuron properly. Only then you will be able to understand the name and what is going on, what is the mechanism. For that you can go check out my previous video. Okay, so see, now we're going to see the exodendritic. Okay, this is the most common type of synapse. So see here, this is the pre. Okay, this is the pre and this is the post. So this is the synaptic neuron, and this is the postsynaptic neuron. So axodendritic, what does it mean? Axon of this pre-neuron, or what you call it, presynaptic neuron, and the dendrite of the postsynaptic neuron are involved in the synapse transmission. So this is called as the exodendritic transmission, and this is the most common type of transmission, synapse transmission. Okay, now we're going to see what is the exosomatic transmission. Exosomatic, by the as the name suggests, the pre-synaptic neuron का axon वाला part and the somatic part of the postsynaptic neuron. So see, this is the synaptic part, so this is also common. Okay, not as common as the exodendritic, but it is also common. So that is the exosomatic. Now we are going to see the axonic. Axonic, it is the presynaptic neurons axon and the postsynaptic neurons axon. When they are involved in the synapse transmission, it is called as the axonic. It is not as common as both of these. So see, the most common is the exodendritic, then after that comes the axosomatic, then comes the axonic, and at the last we have the dendrodendritic, which is the rarest one. Okay, this is the rarest one. So this is the presynaptic neuron, this is the postsynaptic neuron, and both the dendrites of both the neurons are taking part in this. So this is called as the dendrodendritic, and it is rarest. Okay, it is rarest. Now we're going to see according to the nature of the transmission. According to the nature of the transmission, this is very important to know. Okay, this is very important to know. Okay, I'll select this color. So see, chemical synapse, electrical synapse, and conjoint synapse. We have three types of transmission. So say release of neurotransmitter is called as release of the neurotransmitter is the chemical synapse. Transmission of impulse from one neuron to another neuron without secretion of neurotransmitter, but impulses through the gap junction is the electrical transmission, and the conjoint in which both are involved, the chemical transmission as well as the electrical transmission. So this is the second one, and the third one we are going to see is the according to the number of neurons. So see, 1 to 1, what does it mean? One neuron ends on another for the transmission. So it is mainly seen in the neuromuscular junctions. Okay, one neuron ends to the another neuron in the neuromuscular junctions and the parasympathetic, and in the many to one, multiple neurons end onto a single neuron for the transmission, usual type of CNS. Okay, so this is a usual type of CNS. One to many. Okay, one neuron contains many post synaptic neurons. Okay, it's acting on the post more neurons, so that is in the sympathetic. See, 1 to 1 is in the parasympathetic and 1 to many is in the sympathetic, and whatever comes between that, it is the many to 1 that is seen in the usual type of CNS. So these are the types of synapses that we have seen on the according to the part of the neuron involved, according to the nature of the transmission, and according to the number of neurons. We have seen this. Okay, so we have seen the types of synapses. Now we are going to see in the cerebral cortex. See, 80% are the dendrites and 50% are the, oh my god, cell body. Okay, I'm sorry, I didn't understand my own handwriting. So 80% are the dendrites and 15% are the cell bodies. So see, axonic is less and the dendritic is rare, as I've already mentioned it above, right? So it is about that. Now, functional anatomy of the synapse. Okay, the functional anatomy of the synapse, we are going to see that. Without understanding this, we will not be able to understand the further chapters, so make sure you watch this with full focus and full concentration. It is very important. So see, information donor, who is donating the information, is called the presynaptic neuron. I have written this in my language. And the the information receiver, whoever is receiving the information, is the postsynaptic neuron. Giver, who is giving, who is donating, is the presynaptic. Receiver, whoever is receiving it, it is the postsynaptic neuron. Okay, now we're going to see. So see, this is the neuron, this is the presynaptic neuron. As you see here, this is the presynaptic neuron, and this one, whatever you see here below, is the postsynaptic neuron, and the area that is present between it is called the synaptic cleft. So this is the structural anatomy of the neurons. So now we are going to see what all does it contain. Okay, so this is we saw this is the presynaptic and the postsynaptic and everything. So now the presynaptic axon terminal. We are going to see the presynaptic axon terminal. So see, this is the axon. This is the presynaptic neuron, and this is the axon, and this is the terminal. So it contains two things, that is the synaptic vesicles and the dense tufts. Now we are going to see everything, okay, related to this. And this synaptic vesicles, they secrete the neurotransmitters, okay. From the presynaptic neuron, the neurotransmitter are released and they enter the postsynaptic and cause changes, and that's how the transmission takes place. So see, this is the terminal end of the neuron, and it is 1 millimeter in the diameter. It is also called as the synaptic knob, okay. It is called as the synaptic knob. Now we will see the synaptic vesicle, okay. So see, there are three types. There is a large vesicle, a small vesicle, and again a small vesicle, but this is the clear core and this is dense core, okay. So see, the large vesicle, it is present throughout the it is present throughout the terminal, and it is released by exocytosis. We are going to see the whole procedure. We are going to see the whole mechanism, how it is formed, how does it come out, and how does it come in the synaptic cleft, and how does it get how does it bind to the receptors present on the postsynaptic. We're going to see everything, but yeah, just okay. So this is the clear core small vesicle, and this is a dense core. Both of them released into the synaptic cleft through active exocytosis, okay. This is by the passive and this is by the active exocytosis, recycle back into the terminal by endocytosis, okay. When the neurotransmitter is released, these vesicles, they are into the terminal. When it is released, they are taken they are recycled into the terminal by endocytosis. They are given out by exocytosis and they are taken back by endocytosis, okay. They fuse with the endosomes present inside. New vesicle is formed by budding. By which process? By budding process, new vesicle is formed and filling of the neurotransmitter takes place, and again it becomes a new vesicle, okay. It becomes a new vesicle that contains the neurotransmitter. So see, the synaptic vesicles, large vesicle is present throughout and released by the exocytosis. It is done. उसके आगे कुछ भी नहीं है. And small vesicles, clear core and small vesicles of dense core, they are released into the synaptic cleft by active exocytosis. They are recycled back by into the terminal by endocytosis. They fused with the endosomes. The new vesicle is formed by budding and filling of the neurotransmitter takes place. So this is about the vesicles. Now we are going to see what is the about the dense tufts, okay. They are the filamentous projections, okay. They are the filamentous projections, okay. Contains oh, it is getting saved. Yeah, so it is a filamentous projection, okay. Close to it, it contains with the vesicle. It helps in exocytosis of the vesicles near the active zone. We are going to see what is active zone now. Here we'll see that. But they are the filamentous projection. They are close in contact with the vesicles. They help in the exocytosis. They help in the exocytosis of the vesicles near the active zone, okay. They help in the transmission, okay. Now we are going to see about the synaptic cleft. It is a gap between the presynaptic and the postsynaptic neuron. So this is the presynaptic, this is the postsynaptic, and the space between them is called the synaptic cleft. It is filled with the extracellular fluid, okay, extracellular fluid. It helps in diffusion of the neurotransmitter from pre to post, and it occurs through this cleft, okay. So suppose this is the the neurotransmitter, and these are the receptors present on the postsynaptic neuron. So it helps in binding of these receptors and it helps in diffusion, okay, traveling of the neurotransmitter from the pre to the post. So it is providing a medium. So this is called the synaptic cleft. Now we are going to see what is the postsynaptic membrane. So we already saw the presynaptic membrane, so now we are going to see the postsynaptic membrane. It contains receptors for the neurotransmitters. As I have already told, it contains the receptors for binding of the neurotransmitter and for conformational changes. Okay, area of the postsynaptic membrane, it is modified for transmission. Okay, see the area of this postsynaptic membrane, it is modified for transmission. That is called as the prosynaptic density. Okay, the prosynaptic density, it contains cluster of receptors. Okay, whatever the postsynaptic membrane is there, it is modified for transmission, which is called as the postsynaptic density. It contains a cluster of receptors. Now we are going to see what are neurexins. Okay, they are the presynaptic proteins. They are the presynaptic proteins. Okay, receptor for neurexin protein postsynaptic membrane. Okay, so see, they are the presynaptic protein. The neurexins, they are the presynaptic protein. Receptor for this neurexin protein is present on the postsynaptic membrane. Of course, for binding, it helps in binding. Okay, structural which provides structural stability and the synaptic architecture, coded by alpha and beta isoforms. Now this is synaptic specificity. Okay, it helps in specification, does not let any wrong neurotransmitter get on the wrong receptor. So it is synaptic specific, it maintains the synaptic specificity. So this releases a protein, the protein receptor, that is a neurexin protein receptor. It is present in the postsynaptic membrane, which provides structural stability and synaptic architecture, and it is coded by which isoforms? Alpha and beta isoforms. So this is about the neurexins. Now we are going to see the step of synaptic transmission. So see, there are two mechanisms, that is the presynaptic mechanism and the postsynaptic mechanism. So two mechanism, presynaptic mechanism and postsynaptic mechanism. What are the changes occurring in the presynaptic membrane for transmission of the neurotransmitter, and in the postsynaptic mechanism for binding of the neurotransmitter on the receptors that are present in the post synaptic membrane. So we're going to see everything. So see, we'll see the presynaptic membrane first. Okay, it is with the help of docking and priming. Remember that I will talk about this in subsequent topics. So see, attacking of the, here only we'll talk. Okay, attacking with the membrane of the whatever neurotransmitter, attacking vesicles, attacking with the membrane, it is called as a docking. And what is priming? Ready to release the neurotransmitter out of the vesicle, it is called as priming. So what is docking? It is nothing but hitting or attacking of the vesicle on the membrane so as to release the neurotransmitter. So when it is ready to be released, the neurotransmitter is ready to be released, it is called as priming. So this comes under the presynaptic mechanism. Now after, when this happens, action potential at presynaptic axon is generated. Okay, the action potential is generated. Depolarization depolarizes the presynaptic membrane. So the depolarization of the presynaptic membrane occurs, which causes the opening of the calcium voltage gated channels to enter the axon terminal. Okay, through the AZ, that is the active zone. Through the active zone, we saw the active zone, right, जहाँ पे vesicles से neurotransmitter release होते हैं and वहाँ पे gates होते हैं for the transmission. So see, causing opening of the calcium gated channels to the axon terminal through this. Okay, तो what happens पहले से देखेंगे. See, docking and priming occurs जहाँ से neurotransmitter बाहर आता है. Action potential is generated. Depolarization of this membrane takes place, which causes the opening of the calcium voltage gated channels, and influx of the calcium ions takes place. So the concentration of the calcium ions increases, which causes the exocytosis of the vesicle. Okay, exocytosis of the vesicle. I'm sorry, let us understand this with the diagram. Okay, so see, suppose this is here is the active zone. Okay, so the vesicles are present inside. What happens is that the action potential is generated, and the calcium channels they open, and influx of the calcium ions takes place, which causes conformational changes, so that this vesicle comes out and it releases the neurotransmitter in the synaptic cleft, so that this binds with the receptor that are present on the postsynaptic neurons for transmission. So see, we'll see the flow chart again. See, docking and priming, then action potential generated, generated, depolarization occurs, calcium channels open, influx of calcium. What does it do? Calcium mediated exocytosis of the vesicle and release of the neurotransmitters. So see, calcium causes fusion of the vesicle on the membrane by causing microfibrils to contract to help in discharging of the neurotransmitter. That's what I told, right? When the vesicle, it releases the neurotransmitter. How does it release the neurotransmitter? It causes fusion of the vesicle on the membrane. Okay, fusion hota hai and causes the microfibrils contraction होने की वजह से the neurotransmitter, it is discharged, which is called as the kiss and run discharge. Okay, the specific name is given. This is the kiss and run discharge. Now, neurotransmitter discharge in the synaptic cleft passively. It is the discharge is passive. Okay, it is not active, but it is passive. Release of the neurotransmitter is called as the Dale's phenomena. Okay, 200 micro seconds, and which occurs due to the calcium influx. As we already saw, the release of the neurotransmitter takes place, which is called as the Dale's phenomena. So this is about the presynaptic membrane. This is about the presynaptic membrane. Now we are going to see the postsynaptic mechanism. We saw the not membrane mechanism. Okay, the postsynaptic mechanism. So the neurotransmitter binds with the postsynaptic receptor. I told no that the receptors are present on the postsynaptic neuron. So whatever neurotransmitter is released from the vesicles, which is called as the Dale's phenomena, okay, that binds with the receptor that is present on the postsynaptic neuron. Conformational change occurs, opening of the channel or the cascade reaction occurs. It can be movement of the cation or the anion. See, opening of the channel takes place, no? So either it can be a cation or an anion. Both, either it causes depolarization or the hyperpolarization. Okay, depolarization causes excitation and hyperpolarization causes inhibition. We will study that in this video itself. We will study it in subsequent topics. So see, what is the role of the membrane protein? We saw that neurexin is there, no? So what is the role of that membrane protein? Vesicle membrane gets coated with the clathrin. Okay, so there are two types. So that is the synaptobrein and the synaptotaxin, syntaxin. Okay, so it is a V type of protein and this is the T type of protein. It helps in docking and priming. Okay, this one, the synaptobrein, it helps in docking and priming. What is docking? It is the attacking of the vesicle on the membrane so that the neurotransmitter can come out, and priming is nothing but release of the vesicle from that particular region for the release of neurotransmitter. When the neurotransmitter is just ready to come out, that is called as priming. Okay, so this synaptobrein contains the V protein that helps in docking and priming. Now the syntaxin contains the T type of protein. Okay, T type of protein, its function is not given in the textbook. Now what is the clinical integration? The neurotoxins inhibit the release of the neurotransmitter. Of course, this is very true, right? The neurotoxins, they bind to the receptors, okay, and inhibit the release of the neurotransmitters. Okay, no, it doesn't bind, but it does not let the release of the neurotransmitters, it does not let them come out. Now, so this is done. Now we are going to see the postsynaptic mechanism is also done. Okay, the postsynaptic mechanism is also done. Now we are going to see what is the synaptic potential. So see, when the either depolarization occurs or the hyperpolarization occurs, depolarization causes excitation and the hyperpolarization causes the inhibition. So see, the synaptic potential is generated. So this is a very important topic, that is the synaptic potential. Add stars in your notebook as well as in your textbook, okay, or take screenshot. Okay, synaptic potential is generated due to depolarization or the hyperpolarization. Okay, this potential is of longer duration than action potential. Okay, this duration is longer duration than the action potential. So see, it can be excitatory postsynaptic potential, or the inhibitory, that is the helps in inhibition. So it is also called as IPSP, and it is also called as this is called as EPSP, this is called as IPSP. I'm sorry, my bad. Okay, so what does it do? See, the excitatory, first we'll see the mechanism and then it will be very much easy to understand. See, the synaptic potential, what happens is the the action potential arrives at the presynaptic neuron, which helps in opening. We are seeing from beginning again, okay? The action potential arrives and the presynaptic neuron gets excited, opening of the voltage gated calcium chambers, calcium channels, and I already told, no, the calcium channels get opened, due to which the vesicle is thrown out. So the influx of ion into the presynaptic terminal. So what happens, this voltage gated channel gets opened and the ions move inside, which is called as the influx. The calcium mediated exocytosis of the neurotransmitter takes place. Calcium mediated, because due to calcium, opening of the calcium channel and influx of the calcium channel, only then the exocytosis of the neurotransmitter is taking place. So it is called as calcium mediated exocytosis. Now binding of the neurotransmitter on the receptor present on the postsynaptic neuron. Opening of the chambers, again, movement of ions, okay, movement of ions, either cation or either either cation or either the anion, both of which cause depolarization होगा या तो hyperpolarization होगा. Depolarization causes excitation and hyperpolarization causes inhibition. Now see what happens is change in the membrane potential. Of course, when there is movement of ions, there is conformational change in the membrane potential, then generation of the synaptic potential occurs. So remember this flow chart, okay? We saw how the synaptic potential starts. Now we'll see about the excitatory and the inhibitory. So when synaptic when synaptic transmission is towards depolarization, example from -70 millivolt to 60 millivolt, excitability of the postsynaptic neuron increases the other stimulus. So what are the neurotransmitters? There is acetylcholine and the noradrenaline, okay, not norepinephrine. Don't get confused, okay, because I used to get confused. So this is the acetylcholine and the noradrenaline. So this is the excitatory. Now we're going to see the inhibitory. It is called as it is due to the hyperpolarization, decreases other stimulus, and this decreases other stimulus. Now let us understand this with a graph, okay, with a simple graph. So suppose this is the graph and this is the 70 millivolt and upside we have 55 millivolt, and suppose we have here minus 35 millivolt, okay. So this is the reference line. So if it is going upwards, so this is the depolarization. If it is going downwards, this is the hyperpolarization. The depolarization, it causes the excitation, and the hyperpolarization, it causes the inhibition, okay, it causes inhibition. So remember the flow chart. Now what are the ionic bases for excitation? We are going to see the excitation. What is the ionic basis? Opening of the calcium and sodium ions, sodium channels, which causes influx, and potassium closure, okay, potassium closure is cause for the excitatory, okay. Calcium and sodium are getting inside and the potassium is closed, okay. So this causes these are the basis. Now what is slow EPSP? Decreased potassium conduction causes slow EPSP. Now the same in the inhibitory is that the calcium and the potassium channels open. It is totally opposite, right? See, the excitatory is totally opposite to inhibition. So see, the calcium the chloride and the potassium, the channels, they open, and the sodium and calcium are closed. So it's completely opposite, counter, okay. So what is slow IPSP? Increased potassium conduction. So see here, slow EPSP is decreased and slow IPSP is increased. So this is about the types of the synaptic potential, that is excitatory postsynaptic potential and inhibitory postsynaptic potential. So now we are done with the flow chart and everything. Now what is the role of dendrites? Okay, what is the role of dendrite? What are they doing? They are increasing the surface area, okay, for the presynaptic knob. Okay, see this, if this is the axon, see this is the bulging part, okay, this is the bulging part. So the dendrites, what they are doing is they are increasing the surface area for this, and the dendritic spine, okay, what it is doing, neuroglan, okay, it helps in the synapse formation. It releases this which helps in the synapse formation, okay. uh, alteration in the spine causes alteration in the dendritic spine. Okay, alteration in the spine causes long durational potential. Okay, the potential, okay, is the whatever potential is there, no, the depolarization or the hyperpolarization, it lasts longer. And the protein synthesis, the dendrites, they help in the protein synthesis. So what are the roles? Increasing the surface area, release of this chemical causes the synapse formation, causes long durational potential, which due to alteration in the dendritic spine, and the protein synthesis, it helps in the protein synthesis. So we are done with this. Now we are going to see transmission through the electrical synapse. Till now we saw the transmission through the chemical synapse. In chemical synapse, neurotransmitter are released. In electric synapse, it is not released, it is through the gap junctions. So neurons come closer to form the gap junctions. The neurons, okay, they come closer to each other to form the gap junctions. Ions pass easily and directly passing, which causes the activation. Okay, so see, neurotransmitter, what happens, the first vesicle has to come out by the calcium channels, opening of the calcium channels and stuff. Then the neurotransmitter has to be released, it has to pass the synaptic cleft, which contains the extracellular fluid. Then it has to go to the postsynaptic neuron, on which the receptors are present, and then it has to bind and bring the conformational changes inside the postsynaptic, cause inhibitory or the excitatory. So it is a huge process, right? But it occurs indirectly. But transmission of the electrical impulse, it is directly. Okay, it is directly, and easy passing of the ions takes place. So see, what are the difference between these two is that in chemical transmission is unidirectional, and in electrical transmission it is bidirectional. Okay, so see, once the neurotransmitter comes to the postsynaptic, it cannot again go back to the presynaptic. But the electrical transmission, it can come like this, and it can also go in the opposite direction as well. So it is unidirectional, and this is bidirectional. Modifications, it can occur superiorly, but it is not seen in this electrical impulses. It is slower conduction. Why slower conduction? First, the action potential has to be generated, then the calcium channels, they have to open, and then calcium channels, then ions, then again influx of that, and release of the vesicle. From that vesicle, again docking and priming, and the neurotransmitter has to come outside. It has to pass through the ECF, that is the synaptic cleft. Then on, it has to go on the postsynaptic and bind to the receptors and bring conformational changes. So this takes time. It is slower conduction, and this is faster conduction, because directly it can go, because see, the neurons, okay, they are coming very very close to each other, causing gap junctions are present between them, due to which it becomes faster and it becomes easier, and it is direct. So this is about the whole chapter, not the whole chapter. So this is about the mechanisms of the presynaptic, the postsynaptic, how it is transmitted, what are the mechanisms, what are the names and everything. Now I would like to ask you a question. What is the release of the neurotransmitter from the vesicle? What phenomena it is called? Okay, the release of the neurotransmitter from the vesicle, what it is called? Okay, what it is called? It is a dash phenomena. Okay, it is a dash phenomenon. You have to name I want the answer in the comments. Okay, so this is it. Now in the next video, we are going to do the neurotransmitter, the properties of the synapses and the neurotransmitter, and the chapter ends here. So thank you so much for watching.